Welcome to ScienceStars. Topic: Biotechnology. We are going to have a quick revision. Let's get started. Biotechnology refers to the integration of natural sciences and organisms, cells, parts thereof, and molecular analogues for products and services. This definition was given by EFB, which is the European Federation of Biology. Two processes that gave birth to biotechnology are genetic engineering and bioprocess engineering. Genetic engineering involves techniques to alter the DNA or RNA. Bioprocess engineering deals with maintenance of sterile ambience to produce only the desired microbe. Processes of genetic engineering: creation of recombinant DNA, gene cloning, gene transfer. You must know that there is a site on DNA which is referred to as the origin of replication. At this site, the replication process begins, and any alien DNA linked to it can replicate and multiply in host organisms. Also, plasmid is an autonomously replicating extrachromosomal DNA that is circular in its shape and usually found in bacteria. The first plasmid was isolated from the bacterium Salmonella typhimurium by Cohen and Boyer in the year of 1972. Steps in genetically modifying organism: identification and isolation of desired DNA. Introduction of desired DNA into the host. Maintenance of introduced DNA in the host and transfer to the progeny. Tools of recombinant DNA technology. Firstly, we have restriction enzymes that are also known as molecular scissors. They help cut the nucleotides from DNA. Two types of restriction enzymes are known: exonuclease, which remove nucleotides from end, and endonuclease, that remove nucleotides by recognizing specific palindromes in DNA. Now, what are palindromic sequences? Palindromic sequences read the same on two strands when read in the same direction. Example: 5 feet got TC minus 3 feet on one strand and 3 feet CT tag minus 5 feet on the other strand. Rules for naming restriction enzymes. The first letter comes from the genus from which it is isolated, hence written in capital. The next two letters come from the species. The next letter is the strain of the bacteria. And the last one is the Roman numeral in the order of their discovery. You can refer to the example shown here. The second tool for RDT is ligase enzyme, which helps to join DNA with vectors. It is also called molecular glue. The third tool is vector, which is required for transferring the recombinant DNA to host organism by the process called transformation. A vector must have an ORI site with high copy number, selectable markers to eliminate recombinants from non-recombinants, and preferably one cloning site. Important point to be noted that tumor-inducing plasmid of Agrobacterium tumefaciens and retroviruses have been modified to deliver genes to higher plants and animals respectively. The fourth tool is the competent host itself to take up the DNA. Various techniques can be used to make the cell competent, such as it can be treated with divalent cation. Or DNA can be forced by placing the cell on ice, then giving heat shock, and again placing it on ice. Microinjection can be used, by which it is directly inserted into the nucleus. Biolistics or gene gun method can be used for plant cells, by which DNA coated with tungsten or gold is bombarded. Summarizing recombinant DNA technology, we have: in RDT, gene of interest and vector are cut by same restriction endonuclease, and then are joined by ligase to form recombinant DNA, which is then transferred into cell by process of transformation. Important points to be noted: DNA fragments are separated by agarose gel electrophoresis, and DNA, being negatively charged, moves towards anode. Smaller fragments move farther than larger ones. Separated fragments can be only visualized by staining with ethidium bromide and then putting them under UV rays. The separated fragments are cut down from gel by the process of elution. B. Cells have many other molecules other than nucleic acids, so cells are treated with enzymes to digest other molecules, like, for example, bacteria is treated with lysozyme, fungi with chitinase, and plant with cellulase. PCR stands for polymerase chain reaction, which is used to amplify the segment of gene or produce multiples copies of it. The process consists of 3 steps. Denaturation. In this step, the DNA strands are separated by high temperature and Taq polymerase is added. Taq polymerase is a thermostable DNA polymerase isolated from the bacterium Thermus aquaticus. It helps to carry out the process at very high temperature. The second step is annealing, in which 2 primers are joined to specific complementary DNA sequences. Third and the last step is extension, where the primers extend and form the complementary strands of DNA. Important points to be noted: production of recombinant proteins on a large scale requires the use of bioreactors. and stirred tank bioreactor is used to ensure availability of oxygen mixing throughout the entire process. Downstream processing. It includes separation and purification of the finished products. It can undergo clinical trials as in case of drugs.